Hello everybody and welcome back to another PPI lecture review. Today we're going over mechanism of action, dose response curves, drug targets, and toxicology. So let's start with pharmacodynamics. In the last lecture we mainly talked about pharmacokinetics. That's essentially what the body does to a drug. Pharmacodynamics is the opposite. It's what the drug does to the body, and this is traditionally what we're trying to learn in pharmacy school as pharmacists. We want to know what these drugs are and how they work. Um, and drugs don't just do one thing in the body. I think it's very easy, especially with the level of biology we're taught in our undergrad, assuming we don't have some higher level bio from maybe a different degree path before this, that drugs and just things in general that fit into receptors, that fit into enzymes, it's like a lock and key. It must fit perfectly. And that's not quite the case in the real world. And drugs can actually interact with a ton of different receptors in the body. They can have a ton of different responses. Um, and due to this, a drug can have multiple effects. So a few examples of this are, for example, let's say a lot of your psych medications. So think like something like an SSRI, so like escitalopram or sertraline. These affect serotonin receptors primarily, but they also interact with dopamine and a bunch of other things. So again, just similar structures, these drugs can have multiple effects. So just basically something to keep in mind. Um, Now, there's different ways drugs have interactions. A really primary, really basic one is whether or not a drug is hydrophilic or lipophilic. Um, this is one way it can affect interactions, both from if a drug is lipophilic, it might be able to more easily access receptors that are in fatty areas of the body, whereas hydrophilic not as much, or it could deal with the actual shape of the enzyme. For example, if an enzyme's hydrophilic, it most likely has a lot of hydroxyl groups, typically, not always. But with these OH groups, you can form hydrogen bonds to receptors more easily, so that could be a factor. As well as enantiomers, you might remember from organic chemistry that there's different, they call it handedness of drugs, essentially the way that molecules and atoms are rotated around bonds. This can also affect how well something binds to receptors, so that's something to keep in mind. Not as important for this class, but something to keep in mind. Another thing is not all drugs even target receptors. A really good example of this is Tums, which is just calcium carbonate. And literally all Tums is doing is the carbonate anion neutralizes extra protons, acidity in the stomach. So that doesn't target a receptor, it more changes an environment. Uh, so again, drugs don't even necessarily need a receptor, it's more just how they're working in the body, which is what we're focused on with pharmacodynamics. As far as different kinds of chemical interactions drugs can have, there's four main ones. There's van der Waals. These are very weak interactions, but all molecules will have them to an extent. It's basically it's very easy to model electrons in a molecule as always being in one place, but in reality, they're in a cloud. And statistically, electrons might group up into more places than others just at certain time points, and that'll create slight like polarity and charges that might attract drugs. And those are small or instantaneous, but they can have effects, especially on larger molecules that have a lot more electrons that have a much higher chance of something like that happening. Next up is hydrogen bonds. These are quite a bit more common. We did talk about them. Hydrogen bonds will always involve a nitrogen, oxygen, or fluorine, although typically in most molecules it's going to be the nitrogen or the oxygen, although it can happen with fluorines, just that's a little less common. These are bonds just when a hydrogen atom is attracted to these electronegative atoms, nitrogen, oxygen, fluorine. So that can create some polarity. Then there's ionic. This does happen in organic molecules. A way you can think about this, because when you think ionic, you might remember Gen Chem, you might think something like salt, which is Na plus Cl minus. Remember that there's also more organic structures, so you could have a molecule that's, let's say, NH4 plus, then two of those bound to like a carbonate. So even though this is a kind of like more organic system, there are still ionic charges here just based on how these atoms interact with each other. And then finally, the strongest type of bond is covalent. These are your classic chemical bonds. They usually involve a carbon bonding to something, at least in drug molecules, although not necessarily. Could even be something like an oxygen bound to a nitrogen, although that's something like that's going to be a lot less common. It's not very stable, but it is, again, something that can happen. So now let's talk about some of the types of drug channels. There are ion channels. You've probably seen these in either biology or anatomy. A really classic one is that sodium-potassium ion channel that powers up membrane potentials for the cells. These can be gated in some cases. In the case of the sodium-potassium channel you may have learned about, those are voltage gated, meaning that when enough charge happens across the membrane, they'll automatically open. However, I want to introduce a new type today. These are called ligand-gated ion channels. Um, and essentially, these will bind some kind of substance, some kind of agonist, which, to define this, I believe we talked about this in the last video, but an agonist is something that works positively to influence a drug system. It'll elicit a response, where an antagonist will nullify a response, and an inverse agonist will actually create the opposite response to an agonist. So something just to be aware of. So an agonist is basically just something that activates a system. Acetylcholine is a very common one. You'll see that as ACH a lot. And this will come back quite a bit throughout pharmacy school. There's also GABA as another example of this. This one's a little less common. You'll see this later on more so in your like neuro-related classes, but I don't really think you get those until your third year, so you've got a little while, but you might see them a little bit in this class. And then those are open ion channels. So what's moving is you have things like sodium ions, chlorine ions, potassium ions, and those can move in or out of a cell. So that's ion channels. There's also what are called G-coupled protein receptors. You're going to see these a lot. They're a little bit complicated at first, but you will get more used to them. I will try to do my best to explain them right here. These also involve binding of an agonist, or they can. Essentially, an agonist will bind. You have this receptor here. You can see it crosses the membrane. These will actually cross 7 times. They're kind of depicted as a squiggle usually, but you can see there's like 7 of these orange cylinders. That's not incredibly important to know that there's 7. It's just how it is for all of them. So if you ever are asked that, although I don't think you'd ever be, just put down 7. And essentially what will happen is something very interesting is these 7 units of protein are usually bound to other, what are called subunits. And these aren't part of the main protein, but they're linked in some way. You can see this little squiggly right here. Let me erase because I think I'm actually making it harder to see. This little squiggly that's connecting them. What'll happen then is when this ligand binds, it'll actually detach these. And they'll then move on and they'll essentially activate other parts of the cell. And this is what's called a cascade. Specifically, one that's very common is ATP being turned into CAMP. CAMP, if you remember that ATP means triphosphate. AMP is monophosphate, adenosine monophosphate. And essentially what this means is this can tack on a single phosphate group to just different parts of this. So this CAMP can go and regulate things more downstream, or it could come back and activate this system. I don't know specifically what this system is for. It was in the slides, but it is a good model just showing this kind of activity where a subunit detaches and activates a different protein system. And this is called a signaling cascade. In the case where CAMP is used, it's called a phosphorylation cascade. So just something to be aware of. Again, not incredibly important for this class, but just a very important drug mechanism overall. If you ever get like into more specifics of drug mechanisms, you'll definitely see this one recur a lot. This next one is similar, but slightly more complicated because this can involve gene expression depending on the type. So this is called a receptor tyrosine kinase. Essentially what these are is these are called dimers. So there are always 2 separate protein subunits for these. And what can happen is if each one binds a ligand, these will actually change shape and be able to link up. They might link up in the middle here, they could link up on this side, it doesn't really matter. And essentially what can happen is at the bottom we have phosphorylation, so we're adding a phosphate on here. And this can be through CAMP. Or ATP in this case, it looks like, because you can see this ATP, triphosphate, becomes ADP, diphosphate. And so you might be noticing a pattern that phosphate's kind of like our activation ion in the body. It's so high energy ATP that the energy change that adding a phosphate onto another molecule can have can actually change its shape. So that's why we use it so much, because it's so high energy. And then from here we can have more downstream processes. These being activated can maybe interact with another protein. It doesn't really matter. I'll show you a specific example of this. This is called JAK-STAT signaling. This is a receptor tyrosine kinase, but it's a really interesting one. It activates the same. In this case, they actually bind the same ligand on the surface, but they still come together like we saw before. You still get that phosphorylation, but I don't know how well you can see it here, but you see this other protein, JAK, right here. JAK gets phosphorylated, but then another protein is able to come along, which is called STAT. I'll write that down. So this is JAK right here, and then STAT will come along. Now what STAT does is interesting. STAT is a dimer that kind of exists normally in the cells, but when this system right here, this tyrosine kinase system gets phosphorylated, they actually take one of those phosphates off for themselves. If two STATs meet up, they actually bind together. They use their phosphate groups. They use those high energy molecules to then create bonds between themselves. Then what STAT can do actually is STAT can go into the nucleus of the cell, and it's a transcription factor. It will actually increase whether or not it can increase or decrease depending if genes are expressed in the cell. So what proteins the cells make, what genetic information is getting expressed. So this is a really powerful protein. And the potential therapeutics of this could be used for even gene therapy if we're able to like create specific cascades with this. Because we could potentially, with a custom stat, we could create specific things to express more of a protein and perhaps someone with a disease. So really interesting there. So that's just a subclass of tyrosine kinase receptor binding. The final major type of receptor are intracellular receptors. These are proteins that just exist inside the cell that wait for a molecule, a ligand of some sort, to cross the membrane and bind with them. These are a little less common. These are also used in transcription a lot, and usually the reason why is it's very hard for most molecules to cross the cell membrane on their own without a channel. So what these usually are is these are usually steroid hormones. Since these are very lipophilic, they can cross right through our plasma membrane. And think of things like testosterone, estrogen. They'll express different genes, especially sex-related genes. So these are able to easily bind and activate these pathways. So that's kind of how they work. So the body kind of recognizes that since these molecules can get into the cell so easily, it can be beneficial to have them regulate different genes based on what the body is in need of, based on our hormones. So really quick, concept called biased signaling. Like I mentioned before, molecules can bind to different receptors, but their effects can be different based on the receptor itself. So one molecule can elicit multiple different responses. Um, this also goes into concept of like selectivity and specificity, which I'll get to in a second here, but think of it in the sense that again, think like an SSRI. Well, it typically has effects on serotonin. It can also affect dopamine, just to a lesser extent, because they're really shaped for these serotonin receptors. But since there's similarities between serotonin and dopamine receptors, they also have a little bit of overlap into dopamine, just less so. Less so than like a full dopamine agonist might have. So next up, I want to talk about these definitions. Selectivity is the number of receptors a drug can bind to in the body. So going back to our SSRI example, our selective serotonin reuptake inhibitor, um, it can bind to serotonin receptors, dopamine receptors, I think in some cases GABA receptors, but I think it's a little less common. So don't quote me on that. But anyways, you have like, let's say it's selective just for the sake of the argument to those three types of receptors. It is selective for 3 receptors. But specificity is a little bit different. Even though it can bind to all these receptors, it typically only has one response. So when we think of signaling molecules in the brain, serotonin, dopamine, GABA, all of these being increased usually lead to feelings of more relaxation, less anxiety, more euphoria. So even though SSRIs aren't necessarily the most selective drugs, they are specific because their only major effect, at least not counting adverse effects, although adverse effects do count in this way, it's just SSRIs, they have a whole host of side effects, so I'm just going to ignore them for the sake of the argument. Let's just say in a perfect world, SSRIs only were to give those euphoric effects, then they would be highly specific. Um, yeah. Oh, and they also bind to acetylcholine receptors, SSRIs as well, maybe less so than GABA. Um, Another example is cholesterol medications like statins. They will target a single enzyme. So statins will, you'll learn about this more in therapeutics. They target HMG-CoA reductase enzyme. So they are selective. They are only selective for this one enzyme, but they are not very specific in the sense that they will lower your low density lipoprotein, for example. They will raise high density lipoprotein. They will lower triglycerides. They can have side effects such as myalgia. Like all these different effects from this one drug is essentially what makes them a high selectivity drug but a low specificity, whereas uh, SSRIs were a low selectivity drug or a high selectivity drug. Well, low or high depending on how you look at it. I guess it would technically be low because they're not very selective, but they are highly specific in what they do. So that's all those definitions basically mean. All right, so I did mention this term earlier. I don't know how much I went over it in my last video, but I do have a section devoted to it right now. These are dose response curves. These are essentially a way to easily model how a drug will affect a system. So an agonist will always elicit a positive response on what it's acting on. And a positive result can mean a lot of things, because an agonist, for example, could target one thing, if you have like an alpha-1 receptor agonist, to raise blood pressure, right? Because you'll learn a little bit about this later in this course. But those alpha-1 receptors will constrict your blood vessels. That'll raise blood pressure because there's less volume for your blood to go, raises the pressure. However, let's say we have a beta-2 agonist. This is still an agonist. However, these actually dilate blood vessels, so it actually lowers your blood pressure. So it's not so much whether it's like raising or lowering a system in your body. It's just an agonist is always influencing its drug receptor to do what it's supposed to do. An antagonist, on the other hand, will essentially block a receptor from doing what it needs to do. It won't outright reverse the effects. So, for example, an alpha-1 agonist, the one that will constrict your blood vessels typically, if it is an antagonist, it won't do anything. It'll just keep the blood vessel the same size and prevent it from changing, is more than anything. So let's say you have a mix of an agonist and an antagonist. If an antagonist binds and an agonist is then unable to bind, um, then there'll be no response. You won't be able to have that change. An inverse agonist essentially will do the opposite to what an agonist will do. So let's say we have an alpha-1 agonist, right? Going back to that example, constricting those blood vessels, raising that blood pressure. An inverse agonist would be something that works essentially doing the exact same mechanism, which I don't know if there is an exact reverse of alpha-1, just to put that out there. But essentially what it would do is it would bind to that receptor, but it would do the exact opposite. Based on how it's shaped, it would actually expand those blood vessels and lower blood pressure. And this is different from the beta-2 agonist in the sense that the beta-2 is a different receptor. So those are just two different types of agonists. It's not the inverse agonist because it's working on a different system. These are a lot more rare because to have something bind to the exact same receptor and have the exact opposite response is not very common. So you'll typically only probably hear about agonist and antagonist, either moving forward that response or stopping that response altogether, but do know that they do exist, just in case you get a question on it. Now let's talk about potency versus efficacy. So potency is essentially the effect that a drug will work at, what concentration a drug will start working, or start working to its maximum potential. And the way that we can measure this is, or first I'll talk about efficacy. Efficacy is the max effect the drug can have. So potency is at what dose the drug will work best at, and efficacy is how well the drug can work overall, like what's its maximum. So I do have an example down here that hopefully models this pretty well. So over here we can see that these are drug concentrations. Something to note about dose response curves is on your y-axis you have your percent response. So this is like how much response you can get over like overall, like how effective is this drug. So it's basically on this side, this is the efficacy, just right F right there. And this is the dose. So this is potency when you're looking at this. So a drug with high efficacy, it will have the highest peak. So you can see here that when it comes to pain reduction, because that's what this graph is measuring, aspirin has the lowest efficacy. It's not as high as morphine, and it's not as high as fentanyl, which kind of makes sense, right? These are opiate drugs. At least morphine is. I'm pretty sure fentanyl is as well. Um, but aspirin's not, obviously. So it's weaker. It has a lower efficacy. You can see that fentanyl and morphine are pretty close in efficacy, but morphine's a little bit higher. The benefit to using fentanyl, though, so you can see you need a much lower dose. While these have very similar efficacy ratings, fentanyl is about 100 times more potent. If you notice down here, this is a log scale, so it goes up by powers of tens. It goes from 0.01 to 0.1, 0.1 to 1, and etc. And these are typically how they're measured. Up here is what a non-log scale looks like. You can see you have this decreasing curve. And while this is nice, it definitely is like nicer to know because our brains don't really work in logs naturally. It's much easier just to see numbers going up by increments. This is a lot less effective to compare drug responses because you can see over time that this tailors off. And versus on this graph, we can see kind of this logarithmic curve, which actually makes it much easier to read and especially compare. Because the most useful thing about a dose response curve is how you can compare one drug response to another drug response, which is really like useful in drug research. Um, so anyways, fentanyl has a higher potency of about 100 times than morphine, just because it starts to go up a lot sooner. Now, where do we determine the point that it goes up sooner? Because you can make the argument. You can see that these tails probably trail all the way off down to the bottom here on both sides. Where do we determine where these drugs are measured? Where do they meet? That's called the EC50, which is the effective concentration 50. This is an estimated value, and essentially what it means is this is where the drug is assumed to have 50% of its maximum response. Um, the reason we do this is because all drug curves pretty much look like this. Um, this would also be called the inflection point on a logarithmic graph. And it's basically where the greatest range of change occurs on the graph, just because the slope is so steep versus the slope not being steep on these places. So this also is the range we like to choose, because this is usually where we'll try to dose drugs ideally as well. So if there's a patient that's more sensitive to a dose, when we lower a dose, it'll have a much more significant effect than when we were at one of these tails. And that's not incredibly important. That's more from like a drug design and development standpoint. But just note that we compare drugs directly at this EC50. So for fentanyl, that'd be about right here. For morphine, that'd be about right here. And aspirin, that'd be about right here. And this is a really good way to compare potency because you can see all the way over here is the distance between these two, and all the way over here is the distance between these two. Efficacy is more just the height. So that's pretty much basic definitions as far as that goes. Um, let's also talk about partial agonists. So partial agonists can't really exist in a vacuum. Truly, there's only agonist and antagonist. A partial agonist needs to be compared to another agonist, essentially, in the sense that it is only partial because it produces less of a response than a different agonist. So let's take a look at this graph right here. So we have our butyl. You can see that its EC50 is first, and its curve is the highest. So not only is this the most efficacious drug on this graph, it is also the most potent. But you can see that while it produces a positive response, these other drugs also do. This hexyl, this heptyl, and this octyl, just less so. You can see that their potency and efficacy kind of goes down. So these would be partial agonists and related to butyl. So hexyl, heptyl, and octyl are all partial agonists of butyl. However, if you're looking at octyl, octyl doesn't have any partial agonists, at least out of these. It is a full agonist, just if we were comparing octyl to something else. But if you're comparing two drugs, the one that has less of a response is the partial agonist. A little bit of a confusing concept over here. I will try to explain it to the best of my ability. Let's say we're administering a partial agonist and an agonist at the same time. You could also technically do this with an agonist and antagonist, but we'll start with partial agonist and full agonist. Let's say if we have because we can only have 100% of something, right? This is percent of binding. So if we have 100% of our full agonist, we have 0% of our partial agonist, because that's just how math works. Then if we have 50% of our full agonist, we also have 50% of our partial agonist. 50 plus 50 is 100, right? If we have 75% of our full agonist, we only have 25% of our partial agonist. That's just concentrations. That's not where this gets confusing. Just know that these add up to 100% no matter what you are in the graph. The reason these curves are shaped like this doesn't really matter. You could technically draw this graph, at least right here, with straight lines. But I'll show you why we don't draw it with straight lines in the graph beneath it. So these curves are essentially the same as before, right? We still have our 100% up here and our 0% down here. They're shaped a little bit differently, but we have like our 0% up here, our 100%. But what happens here is the difference is the response. So this on this graph isn't actually concentration, it's response. So if we have 100% of our full agonist, we have 100% response. If we have 0% of our full agonist, however, you can see that it's not up here anymore. Instead, it's down here. And why is that? Well, it's because it's a partial agonist. It only has partial response. So you can see if we only administer partial agonist, this is 100% concentration of our partial agonist. But you can see it's not 100% response. It's only about, like, let's say 40% of a response. And that's just because this has less of effect than our full agonist. So when this is at a concentration of 0 and this is at a concentration of 100, the response will be 40%. And just something to note is you can usually tell what your partial and your full agonist is based on the fact that the concentration of the one that's at 100% will always have 100% response. So if you're ever asked to identify which one is a full agonist, which one's a partial agonist, look for the one that has 100% response, and that curve will be the one that's your full agonist, and then your other one will be your partial. And then this curve right here is your total response. You can see down here that it's the same, and up here it's the same. These are like the two intersection points of the total response. Why is that? So if we have 0% of this, our partial agonist, and 100% of this, our total response is just equal to 100%. It's our full agonist response. Whereas down here, this 40%, the reason this links up again is we only have our partial agonist, it's fully partial. So our response is just what the partial agonist would like to be too. These places in the middle, everywhere that's not these 2 points, are basically just an average based on the percent concentration of drugs. And this can be made a little more difficult in the fact that like this model up here kind of assumes that your full agonist and your partial agonist will have a very similar potency, which again in real world isn't ideal, but just for the sake of modeling this, it's not too important to keep that in mind. So yeah, that's partial agonist. Let's see if there's anything else I wanted to talk about on this slide. Let me check my notes. Okay, yeah, no, I think that's pretty much everything I wanted to talk about. This is another example of partial agonist. So this doesn't have the dose response, but you can see that buprenorphine is more potent due to that EC50. This EC50 is over here. This EC50 is over here, so it's more potent, but it's less effective. However, there is a period right here where it's more effective, so that's another thing you can keep in mind. If you only need a certain level of effect, you can actually choose, like, because buprenorphine is a partial agonist of morphine overall. However, at this concentration, it's actually more effective. So just little things like that to keep in mind. You probably won't get a question like that on your exam, but just in case you do, I want you to be aware of it. Now let's talk about drug interactions with each other. So there's additive drug effects, which are essentially drug A plus drug B creates a response that's equivalent. So let's say that drug A, this isn't realistic, will always lower blood pressure by, like, let's say 10/3. Let's say drug B will lower blood pressure by like 5/2. The additive effect of these is just a blood pressure decrease of 15/5 if you combine them. That's pretty good. Let's use that example again. Let's say we have drug A lowers it by 10/3. Say drug B 5/2 again. Also, this isn't how you add fractions, obviously, but I'm just saying for the sake of blood pressure, which is systolic over diastolic, let's just assume for this problem that it's not, yeah. So you'd think that it'd be your 15/5 again, right? 10 plus 5 is 15, 3 plus 2 is 5. In this case, it might actually be different. We could have something like 20 over 10. So in some ways, this is called synergy. Synergy is when, in some way, whether this be through pharmacokinetics, inhibiting the absorption or the metabolism of another drug, it can be pharmacodynamic, maybe these react in such a way or activate a complex which benefits this drug. Their effects are actually greater than the sum of their parts. So the 20 over 10, systolic over diastolic, is a different, like it's obviously higher than 15 over 5. Now let's talk about potentiation. So essentially to potentiate, one of the drugs needs to have no effect of its own. So this would be in the case, let's say in this case, drug A doesn't change blood pressure. Let's say it's 0 over 0. And let's stick with our 5 over 2 of B. What a potentiation drug would do is it would make that final response something like 10 over 5. Even though this didn't contribute anything, it again either inhibited the metabolism of drug B or did something to where drug B was able to work better for some reason. But drug A itself didn't do anything except make drug B better in one way or another. And again, those mechanisms and how it happens isn't the most important. Just know that it can. Let's talk again about inverse agonist. I actually have a graph on this slide just because up until this point I've been describing it. So this is our full agonist, right? Our full response. Down here is our partial, less than our full agonist, because remember, that's the criteria. A partial agonist is always going to be less than the full agonist. And nothing can be a partial agonist on its own. It always needs to be compared to a full agonist. An inverse agonist will produce the opposite effect of a partial agonist or a full agonist. So you can see that it's actually producing a negative effect. And remember, that needs to work on the same receptor type and do the opposite thing. Otherwise, it's not an inverse agonist. It's just another drug class. So let's say you're treating a patient, right? And let's say we'll just say blood pressure for the sake of the argument. A drug that we really like to use, but let's say it's out of stock. But let's say we do have a partial agonist that we can use. It's like, we can use it, it's not great, it maxes out at a lower value, but it's still usable. We would never use an inverse agonist because that'll actually produce the opposite effect. So let's say again, it's a drug to lower blood pressure. An inverse agonist would raise blood pressure in some way, which is obviously not good in most patient populations. Not always. You could have like emergency hypertensive crisis or hypotensive crisis. But again, generally we think of blood pressure as being pretty negative as it can lead to organ damage if it's too elevated for too long. So you'd never give an inverse agonist to try and treat a condition that you were originally treating with an agonist, but you could use a partial agonist if you were desperate for one reason or another. Now let's talk about therapeutic index and therapeutic window. So therapeutic index can be displayed by a curve. Everybody's genetics are going to be a little bit different to how they respond to a drug, and that's kind of represented under this bell curve, right? This will be the therapeutic effect. We have here our EC50. Our EC50 is usually modeled off what the population responds to, what they get their 50 response of. So you can see down here, right down the middle, we have our therapeutic effect. This is where most people fall. Then some people may be a little more sensitive to the drug. They might get their effect a little earlier on. Or some people might be a little less sensitive to the drug. They might take more of that drug for them just based on their genetics, how they metabolize it in order to see benefit from that drug. Now, this can be problematic, especially for the people on the higher end of the spectrum, in the sense that there's also something where there is a therapeutic effect. There's also a graph for toxic effect. Drugs obviously can have side effects, adverse effects. You can see that sometimes these will overlap. So if you're in this tail of these bell curves here, and you need this higher dose, you might also be experiencing toxic effects. Now, since this is a lower side of the toxic effects curve, statistically, you're less likely to encounter toxic effects there. But let's say this bell curve was drawn a bit differently. Let's say it was something more like this. Just assume that's a bell curve. You can see here that before where it was only like 10% of people getting affected, it's all the way over here. It's like 40% of patients might experience a toxic effect. It can get really crazy. This is more common with like chemotherapy. Say all the way over here. All of a sudden you have people in the lethal effect, which means this could be a deadly drug at this concentration. If we have like 20% of people in that range, we need to be very careful of the doses we give people. And again, these are based on ED50 for effective dose 50, TD50, which is toxic dose 50, or LD50, which is lethal dose. These two are pretty much interchangeable. I don't think we got any questions where it like asked for one or over the other because they essentially mean the same thing, right? We don't want our patients to die, obviously, but we also don't want them getting just poisoned by drugs either. So we just generally try to avoid the toxic dose whenever we can, and then obviously severely avoid the lethal dose. Um, so now let's go a little more into antagonists. I know I've mentioned them, but I think it really helps to see some graph. Uh, so there's two types of antagonist. There are competitive antagonists, which are reversible. And that's modeled in this graph right here. And something that you really should take away is that the way to reverse a competitive antagonist is dosing. And what do I mean when I say dosing? I mean like raising the dose of an agonist or finding a way to lower the dose of an antagonist. And you might think, why would we ever give an antagonist an agonist? Realistically, we probably wouldn't. Um, it would more be from there's another substance in the body that is similar enough to our agonist where it could bind to the same type of receptor we're trying to bind to and blunt the response. So with these competitive agonists, you can see that the efficacy, our ED50 is right here, our effective dose 50, but it's all the way over here with our agonist. Why is that? Well, you can see that the effective dose, or the maximum efficacy, is the same. And the reason for this is essentially we have so much agonist over here at these concentrations, once we add more, this area under this curve, that we've essentially made it statistically unlikely for an antagonist to bind to a receptor before an agonist. Or it's just basic diffusion, right? If we have so much agonist trying to get these receptors and only a little bit of antagonist, more agonist will be in that receptor eventually. So we're kind of overpowering that response from the agonist. And these are usually the antagonists, the reversible bonds, right? It's usually like hydrogen bonds or van der Waals interactions. A different type is non-competitive antagonist or irreversible antagonist. What will happen is these antagonists will form, let's say, a much stronger bond, like a covalent bond to this receptor, a bond that is, for all intents and purposes, unbreakable. Covalent bonds can be broken, but it's much harder than something like a hydrogen bond. All of a sudden, we see something interesting. So we have our just agonist alone, our ED50, it's the same as up here, right? But then our ED50 is the same with this type of agonist. Well, why is that? Since this is irreversible, essentially, however much antagonist is added to this will be bound essentially forever to its site. That means the agonist can only fill what's left. So these curves are going to have the same ED50 because they still have the same maximum 50% response, right? Where 50% of the agonist receptors are filled. However, the agonist will be filling the other receptors, which will mean their response is always going to be less overall. And since this is permanent, there's no way to reverse it. So an irreversible antagonist will always have a lower efficacy than a competitive antagonist, which will have the same efficacy, but you need a much higher dose of agonist, if that makes sense. And then just another thing, another different type of agonist-antagonist interaction is you have competitive. These are where they bind from the same site. This is what these two graphs assume, is that these are both competitive agonists because they are both binding the same site, and that's why this response is lowered. However, there's also something called allosteric binding. That's when you're not binding at the active site. This can also occur. Let's just say that an antagonist binding right here to the site that's not the active site, because remember, this is the active site right here. If it binds there, it might change the shape of that protein and prevent an agonist from binding that active site. So I guess technically this graph right here could represent, this would definitely represent a competitive agonist, but technically the non-competitive could represent either this or what was I saying? It could either be allosteric or it could be competitive, just depending. But that's essentially that. So antagonist will just always lower that response. It's just if the bond is reversible is essentially what dictates if the antagonism is reversible. Again, here's a little bit more on toxic and lethal dosing. This area right here is called the therapeutic window. These are the differences in the ED50 of the therapeutic effect. And this is where it gets a little confusing. This is called essentially, I believe it's the tolerable dose. Or the toxic dose. You can see here we have a little bit of toxicity, This is what we consider an acceptable amount of toxicity. So let's say that's like a 15% toxicity, or a toxicity in 15% of patients. Now, this would normally be like if the toxicity was like death or like organ failure, we wouldn't do this. But if it's something like muscle soreness or a dry cough, we would take this risk because the therapeutic benefit of this drug to get all the way to this higher concentration is so much more beneficial than somebody like ending up with a cough, which is not ideal, obviously. We don't want to get our patients to have side effects, but if it's the difference between our drug working and not, we do have a little bit of acceptable toxicity. And this is called the therapeutic window, the difference between the ED50 and the acceptable toxicity. Something else is therapeutic index. This is essentially our toxic dose over our efficacious dose. This will always be like a multiplier. In this case, let's just say our toxic dose down here is 4. Our efficacious dose is 2. 4 over 2 equals 2. So essentially, we can double the amount of efficacious dose we have before we reach a toxic dose. But let's say, for example, we deem that this drug was way too toxic with its adverse effects. Let's say its tail came down here and we just hard cut it off at 3. It's like we can't have even 1% of our patients being poisoned by this drug. It would be too bad. That would change it then. We'd have 3 over 2, which would only be 1.5. We could only have 1.5 times the amount, the effective dose, before we deem this drug too dangerous to do. This also works the same with lethal dose. It doesn't really matter, again, toxic or lethal, but just understanding that therapeutic index is the multiplier between toxicity and efficacy, and the therapeutic window is kind of the range in which we can accept toxicity and efficacy. And with that, thank you so much, and I'll see you in the next video.